that's what the foundation is about. As I said, I do not know what I'm doing running a foundation, so I'm open for criticism. If there are ideas, if there are initiatives that families want to see, or doctors and researchers want to see, please let me know. I am more than all ears. So with that, we're going to have a little bit of a change in our presentation order. Professor Petrou has to travel this afternoon, so he's going to kick off. Professor Stephen Petrou is the director of the Florey Institute in Australia and the head of the ION Channel Human Disease Laboratory. Steve's team is really on the bleeding edge of researching treatment options for this disease and exploring really creative and new ideas to try and help our kids. We are incredibly grateful that he's here today. And rather than go on with bona fides, of which he has many, many, I will turn it over to Steve to tell us about the great research he's doing. Thank you, Steve.
Thanks, Seth, for the opportunity to share a little bit about what we're doing in the lab and beyond. The title of the talk, as you can see, is Precision Medicine in KCNT1. And we obviously got interested in KCNT1 from the first description of it when it was a genetic disorder. I think it was about 2014 was the first paper. And it was a fascinating gene when we saw it. Len had already been working on it, probably one of the only people in the world almost at that time that had been studying this. No one knew what it did really. We know the gene was in some electrical organs of some fish, and it was having a very specialised role in the physiology. But other than that, other than Len's work, we really didn't have much of an idea what it did. We certainly found out very noisily that when it's broken, it can have devastating consequences, and that's been obviously what's been motivating us and a lot of other groups to try and understand exactly how it's broken and what the best way to fix it is. And I'll talk about one approach. There might be some more nuanced things around the function of this, and Len there's got some very elegant ideas about that, and that'll be very handy to understand that aspect. From our perspective, and this is sort of just a bit of a overview of how KCNT1's looked at, but also the lessons we can learn when we study other rare disorders that are driven by mutations in other channels and other proteins in the brain, SINGA, SLC6A, and so on and so forth. And where the field started, here, about 20, 25 years ago, really. Prior to that, they knew that epilepsy could be inherited when they looked at twins. And then they started looking at inherited forms of epilepsy in families. And that started to show us that epilepsy was a genetic condition. And that work really went on for about 20 years. And it was really only about around 2010 when the technology to enable DNA sequencing got a lot cheaper and more accessible, did we start to realise that there were a lot of epilepsies that weren't inherited, that started what they call de novo, from new. And the first one was really revealed as a major epilepsy syndrome was Dravet syndrome. And that was in the mid-2000s really, that idea came out. And there still was a huge collection of patients that no one understood, even when there was a genetic condition, it was an environmental thing. No one really understood what was happening, and they had a lot of different clinical groupings of these patients. And some of the groupings changed depending when you looked at the patient at different developmental stages. And so it was sort of complex. But with next-gen sequencing, the ability to actually find that single needle in a haystack really opened up opened our eyes to this previously mysterious group of patients that had if you looked at all the epileptic encephalopathies, it's about 1 in 2,000 live births. If you put Dravet and KCNT1 and SCN2A and all the other things together, it's about in that. So that's quite a prevalent disorder, and it's the major cause of neonatal hospitalisations, at least in Australia. I'm sure you'll see that in other areas as well. So, but it's a very important, because we're so confident in the disorder, it's a very important group of genes that you know, we should be able to do something about it because we understand exactly what the cause is. And we never had that comfort before. We sort of knew, well, there was a gene that was sort of transmitted through a family, and most people that had that gene with that mutation had a disorder, but not everyone. There was a lot of, you know, it was a lot harder to actually think about this concept of developing therapies specifically for that. But I think now it really is an incredible opportunity. And what you'll see here is, as always, there's a genetic component, but there's always what we call acquired, whether that's environmental, that sort of merges and mixes in with the genetic signal and can modify it. You see less of that modification down in the sorts of disorders that we're looking at today because they're so strong that you can't really easily modify them. But I'm sure there will be some cases that appear. And this overall, these are the sorts of things, and it was interesting when Seth was talking about some of the things you're doing. These are the sorts of things that have all come together to enable us to enter this space. You know, years and years of wonderful clinical phenotyping. I mean, and this is sort of in this area was the sort of the vision of Dan Lowenstein and his colleagues to form this EPGEP consortium where they just had a lot of patients who were very well phenotyped, who were ready for when the sequencing technology caught up. And that was exploited in some of the first studies that started to show us the genetic makeup of these disorders such as KCNT1. So the genetics is great, the clinical phenotyping is you know, never been better. We're starting to understand, and the work of groups like Len and other labs are starting to tell us when you do break the gene, this is how it impacts the protein, this is how it impacts the nerve, this is how it impacts the network, this is how it impacts the entire brain. And all of those things are really important when you're thinking about a therapeutic approach. You know, from a clinical perspective, we're starting to see the emergence of data analytics and decision support tools starting to enter the thought patterns. It's a bit early now, but you know, I'm seeing it in my own institute and the hospitals around us about how they're using all the data around a patient, from imaging and genetics, pharmacosensitivity, and trying to make sense of all of that and use machine learning and these artificial intelligence approaches to try and understand what is the best way to think about that patient. This is a critical part. Family groups, natural history studies and registries. As the industry, and you know, there's going to be multiple companies, which is fantastic, looking into this. You know, this is an area that can help all of those companies with their mission to try and deliver medicines. Having knowledge of patients, understanding how the disease looks like in a kid that's born when they're 3 months old, when they're 6 months old, when they're 1 year old, when they're 2 years old. It's really important because if you do an intervention with a therapy, how do you know you're having an effect? How do you know that wasn't just a natural way that the disease panned out? And having that information, being able to access the right patients is really important. So I really implore you to support people like David in their mission to collect these groupings of these patients. And it's a thing that can really help everybody. Our ability to make animal models hasn't been easier. Therapeutic modalities, whether it's a small molecule, an antisense oligo, you know, a cell therapy, there's lots and lots of choices, gene therapy, there's lots of ways to think about it. And also, and as you know, I won't speak a lot about this, but the commercial and regulatory environments are also, you know, very primed to be able to enable things to happen. And we're really looking forward to, you know, these precision diagnostics and therapies to emerge. As you know, with this also, I don't have to tell everybody in this room about this disorder, but I did want to point out for those of you that may not have thought that there are other genes that can give rise to this syndrome of epilepsy of infancy with migrating focal seizures. Clinically, this disorder is driven by these and other genes. So to a clinician who before didn't have access to genetics, would have lumped all these conditions, all these genes that give rise to a similar presentation at the clinic. But what's changed, the fact that we know the gene mechanism now really fundamentally changes the way we think about therapy. Because as a heterogeneous group, a drug that may have helped this patient may not have helped the patient whose disease was driven by another form of gene mutation. And that would have made clinical trials and everything a lot harder. So it is so important, and maybe I'm not sure where the clinical phenotyping and semiology is going to go, but maybe things will start to be attached, like the parent groups are all aggregating around the gene. And that's going to be very natural aggregation, I think. And some genes, such as like SCN2A, have multiple ways that they're broken. So some patients, when the gene is overactive or gain of function, have one clinical presentation. When the gene's broken in another way and they have reduced function, a different clinical presentation emerges. So even within the same gene, you can get clinical heterogeneity. We're not really seeing that. The only source of heterogeneity we see in KCNT1 is some of those patients that have got that really severe form of autosomal dominant nocturnal frontal lobe epilepsy, which is a different clinical presentation. We're not 100% sure of the drivers of those differences that cause that other patient population to emerge, and that will be revealed over time. You'll learn a lot more about this, so I won't go into any detail. Len is the world's expert on that, on that channel that's mutated. One of the ways we study it, we actually use this African frog, and the females have these eggs that when they're born with a whole bunch of eggs, and they can just release a small batch of them every now and again to get fertilised to make tadpoles and more frogs. But we can actually surgically remove some of these eggs, and then we can actually put genetic material in them and they'll make it. So this is one of the very easy and relatively inexpensive ways that we can try and understand, because KCNT1 is a channel. That means it is a pathway for natural ions like sodium to go in and out of the cell. So sodium, potassium, and chloride are natural ions that are in our blood that the body is used that separates them to make little batteries, basically. If you separate charge, you make a battery, and that's the basis of all electrical signaling in the brain. And when that system, that electrical system, is broken, because some of the channels that are underlying that don't work as they should, you give rise to these brain disorders. So we can put the genes into these oocytes. We wait a few days, we inject it, and then we wait a few days, and then we can actually put electrodes into these and directly measure. You apply electrical stimulation to the actual egg. The channel lives in just the membrane, just in this outer part. Like if you think it's a basketball, these ion channels live in the skin of the basketball. We put an electrode in, like the inflating needle, 2 of those in, and then we can measure and control, and you can see these sorts of records. And each of these little records you see like a little grid on a ladder is in response to one of these electrical steps. They're all just shown overlaid on one another, but you actually reveal them one at a time. And simply, the size of this record essentially tells you how many of these channels are active. And we can use the amount of channels that are active and to do all sorts of manipulations to understand how they respond to electricity, how they change when they're mutated, how fast they go, do they slow down, do they speed up, do they change their sensitivity to various things. And that's a way we can characterise them. We've been looking at these for some time now, and there are mutations that we've got in different patients with different disorders, EIMFS, some 18FLE patients, some others where it's been implicated, and also in some cases where we're given samples from physicians around the world, and we end up finding that they're just normal variants. They aren't actually mutations that drive the disease. And it's important to know all those things, because it helps the physicians in the way they think about treating the patient. You get these sorts of records. So this record is what you get from a control or normal copy of the gene. And then you look to see how the mutation changes the properties of this. And as you can see, for the most part, pretty much every mutation makes this bigger. So we call that gain of function. All the green ones are EIMFS. The red one is the 18FLE condition. And you can see not only does it change the size, it can change the speed. You can see here, the signal to tell this channel to open is right here, but you can see it takes its time to actually move up. You can see that's that curve there. In some of these channels, it's almost immediate. And these sorts of things really matter in a system that's using the properties of this channel to control the function of brain. And you can see there's a myriad of changes, and trying to understand what's in common that causes the disorder and what clinical effects it might have when you have a channel that does this versus a channel that does that. It is still being heavily worked on, and we don't have the answers, although Len might have in the next talk, we'll find out. So one of the things that we want to understand is if you have that, and that's an effect at the most fundamental level. If you study the gene, you see how it's broken, and then you want to say, well, you want to also understand how that affects a nerve, and how that nerve talks to other nerves, and how does it affect the network, and how does it affect brain. So you then have to model it in different systems. People around the world have been putting different KCNT1 mutations into mouse models, asking the questions, well, what is that gain of function phenotype that we're looking at here? What does it do when it's in a mouse? How does that mouse compare to the human? And that's important, because if we're going to look at developing new drugs, it's nice to say, well, there's some correspondence between the effect of the gene in the mouse, and if we give a drug and we see a change in the behaviour of the mouse, it gives us a bit more confidence that maybe that same thing might happen in a person. So it's nice to have that correspondence. And so that's part of the reason we understand the mechanism of the disease with the mouse model, and then we also want to know how might we use that mouse model in a cascade to try and deliver a new therapy. And I'll just show you a little bit about one mouse model, this little journey. This was a human mutation. We were looking at P924L. It was made with a CRISPR method. This is that method. It used to cost, the first mouse I made, I had to go to Germany, it was in 2000, it took me 8 months. Or you buy it, it was $200,000. And nowadays, there's a facility around the corner, it's like $5,000 with this new technology to produce these mice, so you barely think about it anymore. Whereas before, it was a big deal. You think about it, and that's, and you study that mouse for 10 years. But right now, it's completely changed. That's how technology is. This P905L is the equivalent position. The 900 is in the amino acid sequence. It's like a string of pearls, and it's the pearl number. And the P and the L are the mutation from a proline to a leucine. But position 905 is equivalent to 924 in the human. So we introduce that mutation genetically, and then we can make the mice start to look at what happens. First thing we noticed, and so in de novo disorders, we have 2 copies of every gene. In de novo disorders, only one of those copies is mutated. It's all, because it's a 1 in a billion chance that that's going to change. The probability that's going to happen in both is so vanishingly small that I've yet to see it in any case. But in the mouse, when you modeled the mouse with a single copy of this broken gene, we really didn't see any disorder. So the mouse didn't, at this level, recapitulate the human condition. When we put 2 broken copies into the mouse, and you just do that by breeding, you have one mouse line that's got one copy, another that's got the other one, you breed them, and then the children have a mixture of either double copy or 1 copy or none. And so you can have a look at that. And so for this particular mutation, you needed 2 copies. And what seems, if you look across the field now, it seems as though the mice aren't as sensitive as the people. So there's a lot of different people have got some mice that even with 2 copies of it have a very mild phenotype. We've got one here that with 1 copy there's no phenotype, but with 2 copies we do get something, and I'll show you. And then there are other groups with 2 copies the mice die, and they can't breed them. And so there's a sort of a bit of a spectrum because of this different sensitivity. Other genes that we've looked at are worse in the mouse than they are in people, and that's just the way it is. It's a mouse, it's not perfect. And you have to understand each of these things are models. The best model of the disease is the actual patient. We try to replicate as quickly, as much as we can through these other systems. When you started to look at the animal a bit more, well, the first thing we noticed that we weren't getting as many. We would have expected to get 25%, 50%, 25% breakdown of the double, the single copy, or the normal. And here we only got 8.8% because these mice weren't as healthy in the womb, weren't coming out, weren't being born. And when you looked at them, they were lighter and smaller. You can see here the ones red, how much smaller they are, and you can see here this is the control, this is the heterozygous mirroring the human, and this is the double copy. And you can see that only with the double copy did you get a sort of an impact on the animal's health. But importantly, when we monitor these cows and basically just have them in their cages and they've got food and water and they're just sitting there, they start to die. After about 50 days, they start to die. And you can see here over a period of about, each of these little blips down is when a mouse died. And you can see here over about 100 days, all of the mice are dead. And the brothers and sisters that didn't carry the mutation were fine. They lived out, they'll live to 2 years, which is a normal age of a mouse, basically. So a big difference in the homozygous animals and the wild types. And when you look at the nerves, actually individually at the nerves of these mice, and you put an electrode on, and you can measure the sensitivity. So normally nerves respond to an electrical input from another nerve. They're all talking to one another, and they're all connected. One nerve talks to another, and it signals by opening up other ion channels and causes electrical activity. And these sorts of graphs tell us, for a given stimulation, how active is the nerve. That's what this is, stimulation level, activity. We count activity by looking, each of these little upward blips is the language of a nerve. It's called an action potential. It's how they communicate. And you can just count the number of these in a period, either get a frequency or a count, and you can see here, for any given stimulation, the animals that have got the double copy of the mutation, their nerves are way more active, and that's something that's entirely consistent with an epilepsy phenotype. So it's something that tells us if the nerve is doing this, and it's important because it gives us clues as to how the system, how you go from a channel change to a nerve property change, and also if you give a drug, you might expect this behaviour to normalise. It would be nice if a drug took the red curve and plopped it onto the black curve, because then that's where you think, well, if you fix the nerve, you hope all the other things that are broken as a downstream consequence of that. And that's what's important. All these conditions have got a lot of comorbidities. Epilepsy, as you all know, is not the only presentation in the patients with this mutation. And there's movement disorders, and there's aphasia, and there's all these other things. Now, it's almost overwhelming if we think, gee, we've got to look at the 10 major things and try to make a drug for every single one of them. Because once it's broken down here, that ramifies through the brain and disrupts the function of a whole host of systems. But if you can wind back and try to fix it at the source, the hope is that all these other things won't emerge, and we don't have to worry about them. Because you can spend a career following the comorbidities in one of these mouse models and trying to understand all the gory details. But if the focus, and that's okay, I think that's, a lot of people do that, and it's important because you get clues as to how that might inform us of another disorder. But if you want a sort of a critical path towards thinking about therapy, you might just want to say, okay, can we go back and fix the consequences of the genetic lesion as soon as possible in that life cycle of the disorder? And this is one of the tests you could use to help you do that. In addition, you can just look at the behaviour of the mouse. I mean, this is the mouse that's got 2 copies, and what we do find is that these mice have spontaneous seizures. Let's see if the video works. And it does. One of the things the animals will undergo is Straub tail. You see that in other disorders. And so it's starting its seizure. Now, this is its brain activity, or EEG. It's starting its seizure activity. And you can see, it's a bit long, this video, I'm sorry, but it's probably worth just seeing how the seizure emerges in the mouse. And it does change. It goes from this phase into another phase in a second. We need a shorter video. All right, you can see now this wild running, this behaviour, you see this type of behaviour in other mouse seizure models as well. It'll do that, and then it quietens down. The EEG will start to go through this post-ictal depression. You'll see a different change, and the animal now recovers. And these animals can have multiple seizures and that's important because if it has one seizure a month and you want to look at the effect of a drug, it's really difficult to quantify. You want enough that if you do an intervention and you monitor for a week, you can actually understand whether your intervention is doing something. So it's sort of important to understand and quantify this. And this is then the quantification. You can see the different early, from day 20 to day 25 on average. These animals were having 3 or 4 of these per day. As the animals get older, from day 35 to day 37, they were having 7 or 8 of these a day. And as I mentioned before, by day 50, between day 50 and day 100, one of these seizures probably kills them. They have such a severe, because they seem to be getting worse, and then one of them, they'll be found dead in the cage. And interestingly, the student who did the work is sitting in the audience here, listen. She noticed that as she was caring for these animals, the animals that had the double mutation weren't, because normally you throw a couple of tissues into the cage and the animals use those to build a little nest, and they sit in it in the corner now with a little bird's nest. But the animals that had the double mutation were just, their tissue was just sitting flat, and then they had no interest in it and they didn't do anything with it. And we can use that as a marker for just general cognitive awareness and behaviour. So you can say, well, do they have the awareness to be able to, it's a very natural instinct for a mouse to do that. And actually, when Melissa looked into it more, you can actually score the quality of the nest. So you can actually use it as a scalable and quantifiable measure. And we can now use this as a very simple, and every single mouse has the little tissue in there, so it's an easy, cheap, and quite robust measure. And so if we do a drug, and remember I mentioned before that we just don't want to stop the seizures, we want to fix the other things. And it would be great if you could fix this behavioural deficit, because it's telling you that you're moving the needle on cognition. So, and that was, so for us it was a really simple but important finding. Another finding was that we have a little big box, it's called a locomotor cell. You put a mouse in there, and it's never been in that before, and they explore it, and the machine just measures how much movement the mouse has in any 5-minute period, basically. You can see a control animal, you drop it in the first 5 minutes, it moved 1,000 centimetres, which is 10 metres. And over time, it got less curious. It's been there, it's smelled every corner, and it eventually starts moving at a, so it's a bit frantic initially, and then it comes down. If you look at the animal with 2 copies of the gene mutation, completely different profile. We don't know what that means, but they explore differently. They move around a lot more, and then eventually, after half an hour, they come back down. I don't know what phenotype, whether this is equivalent to anything in a patient, but what it is a change in the behaviour, again, that if we give a drug, it'd be nice if this behaviour went back towards control. And so our sort of journey in the quest, and this is yet another test. This is basically a big cross that you put up high, and one side of the cross is open, and the other side of the cross is enclosed. And you can imagine yourself up 50 metres in the air, and if you're on the open platform without any handrails, you're going to be less likely to want to stick your neck out and have a look out there because it's frightening. The mice are the same. They're called the open arms. They don't really want to go out there too much because it's scary. Then they stay in the enclosed areas. And you can see here, normally, you can see a normal mouse only spends 10% of its time in the open arms. And they use this test often for anxiety. Animals that are more anxious, or drugs that relieve anxiety, make you go out into the open arms more. And they use this was a test, one of the ways it was used in the pharmacological sense. But when we looked at this, these animals didn't really have any preference, so they would go on the open arms or the closed arms, and it again gave us another measure of altered behaviour that let us think about, well, if we did do a drug, maybe we'd want to see this come down. And overall, we did, survival was reduced, the epileptic phenotype, the spontaneous seizures and intellectual activity on EEG, locomotor cells, so that my movement disorder was different, nesting behaviour, light dark box, and elevated plus maze, all had an element of difference. And we said, okay, now we've got a lot of different things we've got here. If we give an intervention, can we move the needle on each of these? And if we fix them all, is that a sign of a therapy that's got all the right features, that it's going to be a winner when it's given to people? And that's something we're very, very keen to try and help deliver. So, if you face that now, and you might think, well, do you do? We know that now. We know that the channel increases activity. We know the mouse model has got a whole series of deficits associated with it. And then you might think, really simply, because every case was gain of function, can we just make a drug that either reduces the amount of channel or a drug that interferes with the channel and stops it working? So you can either throw the engine out or you can pull the spark plug. This is removing the engine and this is pulling the spark plug. Does the same thing overall, we think. Potentially, again, a lot of these things can be nuanced, but fundamentally, if we can make a therapy that does these, each of these therapies has its pluses and minuses. ASOs are great, I'll talk a little bit more, they're long-lived, but you've got to do a spinal injection every 3 months. These sorts of drugs can be taken oral, but they may not have the right profile that an ASO has. So each approach has its own strengths and weaknesses, and it's good that the industry is trying to converge and work on multiple approaches to try and see what the best one will be. One of the really important things to know when you're going to do this is, maybe before you start or just to have this knowledge, is how safe is it going to be? Because some drug targets, like sodium channels in the brain, if you block too much, you can cause side effects. Not because the drug does something it shouldn't be doing, but even the thing it's doing to the thing you want it to do, if you do too much, that can be bad. And it's nice if that isn't a big liability going into it. And one of the things that we observed is that there's databases of healthy volunteers, more or less healthy volunteers, in a genetic database where they've looked at their gene sequences and had a look whether in any given, all these different channels and any gene in the body, what is the natural variation that occurs there. And what you'll see, and there's about, in the exact database, I think there's like 100,000 alleles in that, but 27 of those patients had at least one, patients of healthy volunteers, had at least one of those copies of the KCNT1 missing. So that means there are people walking out there in the normal, it's about 1 in 10,000 of the patient population, of the world's population, are walking around missing one copy of KCNT1. We know how devastating when you've got a copy of one that's actually broken and working too hard, but we're not seeing anything untoward happening in the patients that are missing one copy. Maybe there's something subtle, but clearly it's not getting noticed and coming up medically and clinically, such that these patients having to be seeking medical intervention. The other line of evidence that you can make a mouse that's missing this gene and you can completely remove it, both copies. So there's 0 KCNT1 in these mice. And these are some of the studies that have made that mouse and studied that mouse. And they've seen some phenotypes when they look. You can see here enhanced itch, sensitivity to a certain type of pain, decreased exploration, impaired spatial reverse learning, so some reduced cognitive flexibility against an enhanced sort of pain response, deficits in some social behaviour. But these are with complete removal of the gene, it still doesn't appear to be as bad as when you add to the function of the gene. So this is good because it gives us a bit of comfort that it's going to be very hard to overdo it. And that's nice to know that going in, especially for the ASO approach, where with a small drug, probably a little bit easier to control that. With an ASO, because you do it and it hangs around for 3 or 4 months, you've got less time to be able to intervene in that. So it's good to have this headroom. What are ASOs? You've probably heard about them a lot at this, from parent groups and Facebook, and there's a lot of talk now. Basically, they're chemically modified forms of the natural DNA molecules that are already in our body. So there's natural DNA, and DNA, so there's 2 forms. There's double strands of DNA, and there's a slightly different version that's got a different little chemical change, and that's called RNA, and that's a single strand. And DNA loves to bind to itself, and RNA likes to have other bits of nucleic acid bound to it. So these molecules are chemical modifications of DNA, and they modify them so that when you give a drug, you want it to hang around your body. You don't want it to be chewed up by the natural enzymes and metabolic systems in the body and remove it. You want it to be stable, so the drug, you're not taking a pill every half an hour. So that they've changed these so that they're more stable. They've also changed them so that they actually interact with the gene more tightly. And that's important because the tighter it interacts, the less of the drug you need. The less of the drug you got, the less opportunity there is for unwanted side effects because it's such a weak interaction, you have to have 100 times as much drug around. That 100 times as much drug can now potentially interact with things it shouldn't. So these chemical modifications were important in making them stable, which is pharmacokinetics, making them effective, which is that they bind and then they do their job. And I'll talk about what their job is in a sec. And also important where they're tolerable, so that the fact that you need less of them, or you can subtly alter the chemical, you can make sure that they don't cause their own toxicity. And so they'll develop that way. Some of the properties overlap because one company developed their own proprietary technology and another company developed its own proprietary technology. But what's happening now is some of this technology is coming off patent. So the company that really started this field is a company called Ionis in Carlsbad, and they've been at it for 30 years, and some of their technology that they developed early, patents have a fixed life, those patents are expiring, so other groups can now start to access this without violating the terms of patents, so that there's going to be more activity because of that, I think, in this space. Now, I'll talk about the modes. Now, these ASOs can do a lot of things, and I won't talk about all of them. If we're going to pursue that simple idea that reducing the amount of the gene or blocking it are the two simple things we're thinking about in the first instance. As I said, there may be subtleties and nuances to that, but for now, this is something that we think if we just reduce the amount of channel or we block it, that's got a good chance of being therapeutic. So these ASOs can do various different things depending on their sequence, but one of the modes that's probably the simplest mode to deploy, and the one that we need for KCNT1, is this so-called GAPMA. DNA, if you've got the original plans of something for DNA, it's almost like a copy of that's made, like a blueprint, and that's called RNA. So you go from DNA to RNA, and then RNA is that intermediate that then the proteins are made. And the proteins are the actual structures that are made from the genetic blueprint, but there's this intermediate called RNA. The drugs, these antisense oligos, act on that intermediate. They don't act on the gene that's in the nucleus, they act on this intermediate, which is good in a way because you're not doing anything permanent with an antisense oligo. Other gene therapies are permanent, and so you have to be very confident that the technology you're using is going to be the best thing at the time you take it. In 3 years, if something else happens, you may not be able to avail yourself of that next generation. ASOs are not permanent. So what they do now, if the ASO has a certain chemical structure, and it's called a GAPMA, it's called a GAPMA because the chemical modifications are not uniform across this molecule. You can see here, and there's only 20 of those little nucleic acids. I mean, we've got billions of these make up our genetic material in our body, but it's only a short run of about 18 to 20 of these that ends up being the ASO drug. And this thing has got a little bit in the middle called the core, and then it's got wings. This thing's, because it's different in the middle and the wings, is called a GAPMA. When that binds that RNA, that intermediate genetic message, it targets it for destruction. So you simply bind and you destroy. This other special molecule called RNA ACE will recognise the binding event and it will chew up this RNA. So you're making all this. So you'll make 100 copies of the channel. You then throw the ASO. Only 10 or 20 get made because all the other 80 ones got destroyed, and you can't use them as a template to make the protein. And that's the simple idea behind these GAPMAs, and sort of the thing you might want to do with KCNT1. Apply it in this way. There are other ways you might want to think about, because when you do that, if this is the patient, because it's a de novo disorder, I said there'll only be one copy. There's always 2 copies of the gene. The other one will be normal. But when you do this other approach, we're actually destroying both of them. And we're hoping we'll take a bit of the good stuff down and also take some of the other one down. And just by reducing both, we might, we get to that point where we see good efficacy. It might be better if you can specifically target the damaged one. So you leave the good copy alone, and you only directly reduce the mutated copy. Now, that's really hard, because every patient, there's hundreds and hundreds of mutations that we've seen so far. It's not possible to make hundreds and hundreds of antisense oligos. It's expensive enough to make one. When you go through all the hurdles of drug development, it's tens of millions of dollars. So it's going to be very difficult to make one of these molecules on a single case-by-case basis. So you can, but what you could do, if you did that, you could make an antisense oligo that did bind. You can see this is the oligo, and it's matching 100% with the mutation, but one of those positions would be a mismatch up here. When you do that, you can achieve a preferential reduction in this copy versus that copy. So you can have an antisense oligo for every 5 or 6 of these that get destroyed, only one of those gets destroyed. So you're biasing it towards the damaged version of the gene. One way of broadening the use, because in addition to the mutation there, you can have these SNPs. So these are, everyone's got, they're called single nucleotide polymorphisms. Everyone's got genetic variation. And a lot of these things don't do anything pathological. They just introduce natural variation into the human population. And if often that SNP will be on the same gene where the mutation is. So, but the good thing is, hundreds of people are going to have this SNP. So you can make an antisense oligo that targets the SNP, and whatever the mutation is anywhere else will come along and get destroyed. So that's a way you can use allele specificity to broaden the case. I'm not saying we need to do this for KCNT1, it may not even be necessary, but there are approaches and there's flexibility enough with antisense oligos that should you need to go down to some of these more specialised applications, there is capacity to do that. What I can say, at a recent conference with Ionis, we collaborated with them using that mouse model. They did a presentation earlier this year. They did a presentation showing that a mouse ASO pretty much fixed almost everything that I showed you before was broken in that mouse, was fixed by the ASO. It lived longer, the seizures went away, a lot of really good things happened to that mouse. So it was a really very satisfying conclusion to that study to show how well the antisense oligo could work. So that's one approach that I think has got very good possibilities for people in this room, because if that ASO, now this is a miracle we developed, that was a mouse version, it's a proof of concept. You make an antisense oligo for a mouse. It's never intended that antisense oligo would be used in a person. It wasn't developed with the rigor that you would use when you're developing one of these for a person. But it just shows you to ask the question, well, if we knock down both the mutant and the wild type, can we fix not just the seizures and the survival, all these other things? And it was quite remarkable how effective that was. But what about other ways of thinking about reducing the function of this channel? I said before that, can you make a small molecule? Now, a small advantage of a small molecule, rather than an injection in the spinal cord 3 or 4 times a year, you take a pill. And that's great. So at one level, if you don't have access to a centre that can do spinal cord injections, or you're in countries where that's even difficult to access, an orally available pill is a lot easier mode of therapy to adopt. So if you can make a special molecule that only recognises the KCNT1 channel, binds it tightly, and does the same thing, stops it from being active, that might also be effective. And I'm just going to show you some very preliminary, not preliminary, just a couple of data slides around a molecule developed by Praxis, called Praxis 2904, that's been designed for that express purpose. It's quite potent. You can see here it's less than 100 nanomole, which is very, very potent. These DPMK, this is metabolism and pharmacokinetics, so that you can see here, it lasts a long time. So you take it, this is just how much drug there is around when you measure over like a 70-hour period, I think. Can't read it. It lasts a long time. That's a good property. You don't want the drug to take it and it plummets after an hour, because then your drug level's going up and down like a seesaw. It's really nice to have stable levels, and you get good. And the other important thing is, when you take a drug, you can get into the bloodstream, but you can't get into the brain. And how much gets into the brain is a really important thing. And some of this data is showing that it can get into the brain and be there where you want it to be. So some of the, if you look at all the criteria, you take a drug, it's nice if it's 1 pill a day, not 3 pills a day. It doesn't cause any other side effects. It's got high potency, it gets into the brain, and it interacts with all the wild type and mutant versions of the channel. And so chemically, it's got a lot of these great properties. And I showed you this data before. Remember, this is from the brains of the animals, where we put an electrode in and measure the electrical activity, and we noticed that the electrical activity in the mutant animal is higher here than the wild type. That difference there is part of the pathology. And as I said before, if you could get a drug that made the red go down to the black, that'd be a good thing. Well, that's what this does. Which one's 2904? Is the blue
Green.
one? The green. So the green one is the animal that's got the mutation. When it's exposed to that drug, it goes back down onto the black line. So at one level, that says we can make that neuron look like the neuron from an animal that doesn't have the genetic mutation. And I think that's a really good start for fixing the phenotype. Because if you ask the question, you have a genetic change, and that genetic change alters the properties of a neuron, and that neuron then alters the properties of the speech system, of the movement and balance system, of the cognitive system. Does it all emerge from? And neurons they speak in this language of, for a given stimulation or a given pattern of stimulation, they output another pattern. That's their language. They hear stuff, they listen in one part of the neuron, they decode, and then they give another signal. And that's the language of the brain. Now, if you're disrupting that language in difference, and brain has got systems that are somewhat separable. You know, the system that controls speech is different from the system that controls movement and motion, and different from the system that controls cognition and memory. So is this a sign that we're fundamentally fixing the thing that's broken? Maybe. And then what we're in the process now is of asking the question, what is this thing now going to do to all those different behaviours that we saw in the mouse? Can we reverse all those wonderful changes we saw that tell us that the drug is having an effect on all the comorbidities and the seizures? And hopefully soon we'll be able to report some very exciting news about that. We've also seen some signs that the drug, when given to a normal mouse, also impacts its epilepsy threshold. So this drug may be able to be used in different contexts. And that's always, that's an example of the lessons that you've learned in a rare disease can be applied to the greater patient population, which is an extra really good bonus for the epilepsy patient cohorts. And we're very excited by whether that will be realised. So in summary, I want to say that functional analysis of mutants suggests a gain of function is a common finding. The homozygous model shows strong phenotype with some overlap with patients and will be an aid for drug discovery, and it always has been for the ASOs that are effective in the mice. Small molecule reverses key neurophysiological defects, suggesting it could have the potential for therapeutic benefit. It's got the signs of a thing that's doing the right thing. There's always hurdles, but it's got the signs of, if it is going to work, this is one of the things you'd want to see, and that's important. And I think it's confident that potential therapies are on the horizon. I mean, we always, everyone's going to be careful, no one's going to overpromise, but things are happening that I think, that you should at least be confident that there are companies and labs that are really, striving to try and deliver some therapies for this really special population of patients. I just want to acknowledge people that did the work. Elizabeth Burbano, who did a PhD after doing her MD degree, did a PhD with us, supported by Melody and Nicola. Robert Hatch did the electrophysiology in collaboration with Praxis. Umesh did all the oocyte recordings. Snezana and Chris helped me with the management and leadership of the lab. And of course, Team Praxis led by Marion Whitman, who's the head of biology, with her team, Chris, Zoe, and Lisa also from the more corporate side of Praxis who helped me prepare the slides, or prepared them for me for the Praxis slides. And as I said, Rob works very, very closely with Marion and Chris on these projects. So I'll leave it there and just thank you for letting me share this with you.